get to introduce our next speaker, who is one of our first out of 2 graduate student speakers, Daniel Kohlhase. He's a past co-chair of this symposium. Daniel is a PhD candidate here at Iowa State University studying under Michelle Graham. He's working in plant breeding with a minor in genetics and genomics. Daniel earned his Bachelor of Science in Crop Science from the University of Illinois Urbana-Champaign, his master's in crop production and physiology from Iowa State University. Daniel's PhD research focuses on studying signaling and molecular response of soybean to abiotic stress using a combination of modern and traditional techniques, including RNA sequencing, virus-induced gene silencing, and grafting. So please join me in welcoming Daniel. And Daniel, you may want to put yours on presentation mode because we're seeing your presentation mode, not your full slide yet. There you go.
All right, well, thank you. That was going to be one of my first questions. Can you guys hear me all right? All right, well, first I want to thank the speakers for inviting me today. I remember when I was transitioning into plant breeding in 2016, watching a graduate student do this, I was thinking when I was sitting in the audience, I would really like to be on that stage. By the time I graduate, I really want to be on that stage presenting, and so here I am. So, as Sarah said, in my lab, we use genomics to study stresses. And today I'm going to be speaking on how we leverage genomics to dissect an early iron stress response in soybean.
Try clicking the arrow on your presentation.
Okay, so as a quick review, we all know that plants require different nutrients to efficiently grow, and we can split these nutrients into 2 different categories. We call them macro or micronutrients, and these categories are based on the quantity required by the plant of each one of these nutrients. And so today I highlighted iron. And as you can see, iron is a micronutrient, which means that the plant requires less iron to efficiently grow. But that doesn't mean that iron is not as valuable as some of these other nutrients. And so what makes, or why is iron so important for plants? When we look at the electron transport chain, we can see that 3 of these components contain or utilize iron in the photosynthetic process. And us being in the business of photosynthesis, we know that being a vital element of photosynthesis makes it an essential micronutrient for productive plants. So what happens when soybean doesn't have adequate iron? Well, we call this iron deficiency chlorosis, and this is characterized by interveinal chlorosis. And on the right, you can see a figure of a 1 through 5 rating scale that we use in IDC research, with 1 being a green healthy plant and 5 being a stunted, chlorotic, and even necrotic plant. And soil conditions where we find IDC are typically high pH, calcareous, and waterlogged soils. And these happen to correspond with soils that we have in the upper Midwest, where a large amount of soybean production occurs. And this is a problem that we really need to study and understand because of the high potential for severe yield loss. So a previous study has shown that each point on the scale, or moving between each point on the scale, corresponds to a 20% yield reduction on average. So there have been 30 plus years of IDC research in soybean now. And what I want to highlight for you today from all this research are mapping studies and gene expression studies. And so for mapping studies, there have been multiple studies that find associations across the genome. And specifically what I want to highlight is a major QTL that has been identified on chromosome 3. And this has been validated multiple times over. In gene expression studies, we typically use Clark and iso-Clark to get a better understanding of the response, a more detailed understanding of this response. And over the years, what we've discovered, or some major results from these studies, are that the response in soybean to IDC is very rapid. Recently, they discovered that they can detect a response as early as 30 minutes after stress. Also, contrary to Arabidopsis thinking, in soybean, we believe that it is a root to shoot signal occurring in response to IDC. And also we have identified patterns of differential expression. So in these gene expression studies, they have looked at various time points. They've looked at hours, days, and even weeks. And what they have found are reoccurring patterns of biological processes that show up as significant in these studies. And we call those the hallmarks of IDC tolerance. And so what those include are iron uptake and transport, defense, DNA replication, and methylation. So last year, a paper coming out of Danny Singh's lab, they performed a GWAS on IDC response. And what they discovered were associations across the genome, including the IDC QTL that I just outlined for you. And in collaboration with Michelle Graham and Jamie O'Rourke, when they looked at the distribution of markers within this region, they found a non-random distribution and were actually able to break up the IDC QTL into 4 distinct linkage blocks. And using the results and knowledge from previous gene expression studies, they actually were able to identify genes of interest that show up in each one of these linkage blocks. So far, the body of gene expression work is suggesting to us that there has to be more than one gene involved with this process. So using that thinking and building off of this GWAS study, what we wanted to do was look and see if there were differences or similarities that we can find in the gene expression between a mini panel of soybean genotypes. Because as I previously mentioned, a lot of our knowledge is based on Clark and isoclark, 2 isogenic lines. And so we can't really assume that this applies to all the germplasm. So how do we choose our genetic material? Well, we selected 18 genotypes from this GWAS panel that I just described, and we chose our 18 genotypes based on 8 haplotypes that were identified using sequences in the IDC QTL on chromosome 3. And then once these haplotypes were identified, we looked for variation in the phenotypic response. So what we're looking at here is a heat map of the phenotypic data collected from this GWAS panel. And you can see that it spans over multiple years in the field, as well as hydroponic conditions. So using hierarchical clustering, we were able to actually separate the group of 18 genotypes into 2 phenotypic classes, and we called them iron inefficient or iron efficient classes. And one interesting thing that stood out to us immediately was that even within the efficient class, we can see a subset of 2 different groups. And so immediately that's suggesting to us if we can classify these by the phenotype, maybe there are distinctions in the responses. Using a principal component analysis, using the same data, we can see that, again, we have 2 clusters of genotypes, and they happen to correspond very nicely to the efficient and inefficient class. So with the efficient, with a tight cluster over here on the left, and the iron inefficient on the right. So in our experiment, we grew these 18 genotypes in hydroponics, all sufficient conditions, until the V1, V2 stage. And then at that stage, we moved some plants into new buckets that were either sufficient or deficient in iron. After 60 minutes being in those new conditions, we harvested leaf and root tissue. After we collected the tissue, we isolated and then cleaned RNA and sent the RNA to the DNA facility to be sequenced. So this is a conceptual graph. And what this is showing you is basically what we do in gene expression studies. So what we do is we look at a particular gene and we look at the effect of a particular treatment. And so in this instance, we're looking at deficient and sufficient iron conditions. So for gene 1, we can see the gene expression in deficient conditions is higher than in sufficient conditions. And in gene 2, we see the opposite, where deficient has lower gene expression and sufficient iron, this gene has higher levels of expression. So what this translates to is we would say that gene 1 is upregulated, and then gene 2 is downregulated. So in this experiment, what we did is we took that concept, applied it across the genome in all 18 of our genotypes and across both tissue types. And so what we're looking at here are the number of differentially expressed genes on the y-axis in both tissue types for all 18 genotypes. And here we organize the genotypes with the efficient on the left in green and the iron inefficient on the right in the black. And so what stood out to us immediately with this graph is that we see a range in response. And then within that range of responses, we can see essentially three groups of genotypes. We see some that have differential expression in the leaves and in the roots, like 1, 2, and 8. We see others that have differential expression just to the roots, and then even like if we look at genotype 14, there's very little differential expression in the leaves and in the roots, in either tissue type. So the first question we asked after collecting these gene lists is, what kind of overlap do we see between these genotypes? Do we see pairs of genotypes sharing the same differentially expressed gene list? And so from this graph, so what we did is we did a pairwise comparison of all these gene lists in the roots and then in the leaves. And immediately what stood out to us was that the roots had a little more overlap than the leaves. But when we look at specific numbers within these pairwise comparisons, we can see that in the roots, there were only 134 genes that were shared by 2 genotypes. So what this is suggesting to us is that each genotype is acting a little differently and possibly using different mechanisms to respond to this stress. So the next question we asked was, how are these genes responding across all genotypes, or what kind of overlap do we see across all genotypes? So what we did is we collected all differentially expressed genes in the roots and in the leaves, and then we asked, how many times does a particular gene show up across our genotypes? And what we discovered was that, again, in the roots, we see more overlap, with up to 17 genotypes having the same differentially expressed gene. And then in the leaves, we see only 5 genotypes sharing a differentially expressed gene. And then again, using the phenotypic clusters or the phenotypic classes, we looked to see which genes are unique, or which differentially expressed genes only show up in an efficient genotype, or which show up in only inefficient genotypes. And what we discovered is that there were a lot of genes that were specific to only iron efficient genotypes. In the roots, we don't see any that are unique to the inefficient. So what this is showing us is that in the roots for efficient genotypes, they are using the same genes, or there's an overlap of genes as the inefficient, but they are using novel mechanisms relative to the inefficient genotype shown in this number. So the next thing that we wanted to look at were transcription factors. And what we're looking at here is within each of the differentially expressed gene lists, we pulled out all the genes that are annotated as transcription factors, and then we graphed the level of expression in each tissue type across genotypes. And again, we separated these by the iron efficient genotypes versus the iron inefficient genotypes. And on the y-axis, we can see transcription factor families. So as we all know, or as we all should know, that transcription factors fall into gene families. And so when we're looking at this graph, what stands out to us are 2 things. One, we see a similar pattern as the number of overall differentially expressed genes, where in 1, 2, and 8, we can see a differential expression of these transcription factors in the leaves and in the roots. Some just show up in the roots, and then others, like genotype 14, only show up or have very little differential expression in either tissue type. So the second thing we notice is clusters of these genes. And so this is relevant to us because if we see clusters, that's showing us that these transcription factor families are targeting specific pathways. And if we see the same cluster across genotypes, this is suggesting to us that the different genotypes could be targeting the same pathways. And the same thought process goes if we see clusters that are unique to specific genotypes. So again, looking at overlap, we see the same pattern where we see more overlap in the roots. We see up to 14 genotypes sharing 1 differentially expressed transcription factor, and then only 4 in the leaves, 4 genotypes sharing a transcription factor in the leaves. And then again, using these phenotypic classes, we can see there are more unique differentially expressed transcription factors that are associated with the efficient phenotypes. And so taking the fact that we see different groups of transcription factors and the different levels of expression, this is suggesting to us that the transcription factors are targeting different pathways in response to the iron stress. So in RNA-seq studies, we use gene ontology or Go terms to really pull biological meaning out of our questions. And so what we did is we used our gene lists of differentially expressed genes, and then we looked for overrepresented Go terms relative to the entire genome. And what we discovered on the left, you can see the distribution of overrepresented Go terms for a particular genotype in a given tissue type. And then again, looking at overlap across all genotypes, we saw more in the roots, more overlap in the roots, up to 7 genotypes sharing an overrepresented Go term, and then only 2 sharing Go terms in the leaves. And we can see when we look at the leaves, this primarily comes from 1 and 8. So what was really interesting when we started to dive into these Go terms is that when we looked at Go terms that were unique for differentially expressed genes that were associated just with the efficient lines, we were able to pull out these hallmarks of IDC response that I mentioned earlier. So we could find Go terms associated with DNA replication and methylation in these Go terms. So another question that we wanted to ask using Go terms was, how are the genotypes responding across, how are all the genotypes responding for a particular Go term? So what we did is we pulled Go terms that were significantly overrepresented in iron efficient lines, and then we looked at how many differentially expressed genes are associated with a particular Go term across all genotypes. And so this is what we're looking at, is that pattern for the leaves. And what we see immediately is a distinction between these 2 blue lines versus the rest of the genotypes here on the x-axis. And then when we change the colors to reflect the phenotypic groups that I keep mentioning, we can see that there are 2 green lines on top, so iron efficient on top, and then the rest of them down below. And so we can see these distinctions between phenotypic groups. And what's interesting about this particular graph is that we do see a pink line in between these 2 groups of lines. And so pink actually corresponds to iron inefficient. So this particular genotype is actually showing us that it's trying to respond like the efficient class, but it's not responding at the same level. So when we look at the same pattern in the roots, we can see that there's a lot more overlap between genotypes. And then again, when we change the colors of the lines to reflect the phenotypic groups, we can see this overlap remains. They don't separate by phenotypic class. But 2 things that I want to highlight from this particular figure are this black line, which shows that it's responding, or we would anticipate this to be an iron efficient line, but this is actually genotype 13, which is classified as iron inefficient. And then the other thing we noticed about this figure is that even though we see a lot more overlap, like in this peak, we can still see peaks of iron efficient lines. So peaks that are unique to iron efficient genotypes. So what this is suggesting to us is that even though we saw different genes in the overlap of differentially expressed genes, this might be indicating that these genotypes are using the same pathways, but different genes. So when we put these 2 figures for the 2 tissue types together, leaves on the top and then roots on the bottom, what we notice, again, is that there's overlap in the roots, and we don't see that overlap in the leaves. And so what our current hypothesis is that there might be something lost in translation between the roots and the leaves. So that's what might be distinguishing the efficient versus the inefficient lines. So the last figure I want to show you guys is a comparison of gene expression. So we're looking at the gene expression of all 17 genotypes, of 17 of the genotypes relative to Clark. So we're doing this because, as I mentioned, a lot of our knowledge is based on Clark and iso-Clark. So we want to see how these genotypes differ than Clark. And what we're looking at are genes within the QTO region on chromosome 3. And we can see based on these colors that these genotypes do have different responses than Clark. What stands out to us about chromosome 3 is this block of blue, and these are actually annotated as PHLH transcription factors, which have been identified as genes of interest in iron response in soybean. And we can see that this block actually corresponds to iron efficient. So this is saying that these efficient lines have a lower expression value for these BHLH transcription factors relative to Clark. And then when we look at homeologs for the IDC genes on chromosome 3, on chromosome 19, we can see that there's a different pattern than on chromosome 3. And again, we see 2 blocks for these BHLH transcription factors, with the iron efficient having lower expression than the iron inefficient. So to wrap up, to summarize my results, we did see differences in the number of differentially expressed genes. We saw very little overlap in our pairwise and overall comparisons of the genotypes in terms of differentially expressed genes. Using transcription factors, we can see clusters of families, and this highlights the regulation of different pathways. Using Go terms, they can show distinctions between the phenotypic groups in the leaves. We saw the distinction between the phenotypic groups in the leaves, and then we saw more overlap in the roots. And then using gene expression profiles, we can look at specific regions of interest and look for differences between those other genotypes in Clark, which is what we know. So my take-home message to everyone today, if you walk away with one thing, is that the initial analyses of these data suggest that there are differences in the mechanism and possibly the rate of response to iron stress within the soybean germplasm collection. And so as a breeder, this is very positive news, because this means that we have a variation to use in our efforts to improve soybean lines. So with that, I want to thank my committee members, Doctors Michelle Graham, Jin-Ming Yu, Jamie O'Rourke, Danny Singh, and Steve Whittam for their constant support. And then past and present lab members of the Graham and O'Rourke lab, Lori Lincoln, Jody Hayes, Michael Morrissey, and then a past member is Dr. Chantel McCabe, as well as my funding sources, the USDA, as well as Iowa State University. And with that, I'd be happy to answer any questions. If you think of something afterwards, I've included my email, so you can shoot me an email, and also a QR code if you want to connect on LinkedIn. Thank you.